Welcome to the deep dive. Today we're jumping into something pretty exciting: precision medicine and genomics.
Yeah, it's all about how understanding our own genes, our DNA, is starting to change everything, leading to, well, truly personalized therapies.
Exactly, and we're looking at how this works from the very beginning of drug discovery through, clinical trials, all the way to how these things actually get made.
Right. We've gathered quite a bit of material on this.
Our mission, as always, is to pull out the really key stuff about precision medicine and these genomic innovations. We want to show you how treatments are becoming tailored, personalized without drowning you in technical details.
Sounds good.
Okay, so if we're talking personalized medicine, where does it all begin? What's the foundation?
It really starts with the human genome, our complete set of DNA instructions. It's quite amazing, actually,
A whole blueprint.
the whole blueprint. And while, we share something like 99% of our DNA, it's that tiny bit of variation that makes us unique. And crucially, that variation influences our health, our risk for diseases.
And presumably how we respond to treatments.
Precisely. Think of your DNA like a very detailed personal instruction manual. Precision medicine is about reading the fine print in your specific manual.
Okay, so how do we actually read that fine print?
That's where genetic profiling comes in. It's the tool we use to identify those specific variations, the quirks in your manual, if you like.
And this profile tells us what exactly?
Well, it can give us really valuable insights. Things like, does someone have a higher genetic risk for a certain condition? Or, really relevant here, how are they likely to respond to a particular drug? It's like getting a decoder ring for your own biology.
Wow. So doctors could potentially have this map before they even decide on a treatment path, choosing the best route from the start.
That's the goal, absolutely. And guiding them along that route, we have something called biomarkers.
Biomarkers, okay, heard that term a lot. What are they telling us in this context?
Think of them as real-time signals from inside the body. They can be genetic markers, sure, but also molecules or even specific cells. They act like signposts.
Signposts indicating what?
They can indicate if a disease is present, maybe how fast it's progressing, or, and this is key for treatment, if a therapy is actually working. They give us objective, measurable data.
Okay, objective measures.
Yeah, for instance, there's a figure in one of the sources from early drug development, figure 17.2 actually. It shows how we can use these measurable biological signals, these biomarkers, to connect the dots between the drug concentration in the body and the effect it's having.
Ah, so it helps find the right dose for that person?
Exactly. Finding that sweet spot based on a measurable response. It's about quantifying the effect.
Okay, so we've got the genetic predispositions from profiling and then biomarkers tracking the real-time situation and response. How is this practically changing treatment? Cancer seems like a big area for this.
Oh, absolutely. Cancer treatment has seen huge shifts. Historically, a lot of anti-cancer drug discovery involved using rodent tumor models. They were useful, definitely a starting point mentioned in the development guides.
Right, kind of a standard approach back then.
But now things are much more refined. A core part of precision oncology is understanding the unique genetic fingerprint of a patient's tumor.
The tumor's own instruction manual, basically.
You got it. So if a tumor has a specific mutation that we know drives its growth, we can potentially select a drug designed specifically to target that mutation. It's a much more focused attack.
Like switching from a shotgun to a sniper rifle.
That's a good analogy, yeah. Hopefully leading to better results and fewer side effects because you're hitting the target more precisely and sparing healthy cells.
Can you give an example of that kind of targeting?
Sure. Take the development of things like paclitaxel-loaded nanocarriers. We've talked about nanocarriers before, right? Tiny delivery system.
Yeah, those little drug packages.
Exactly. So the idea here, discussed in relation to structure-activity studies, is that you could potentially engineer these nanocarriers to specifically recognize and bind to markers on a patient's particular tumor cells, delivering the chemo directly where it's needed most.
So tailoring the delivery system itself based on the tumor's characteristics, that's incredible.
It is. And the personalization doesn't necessarily stop after the main treatment. Another source mentions detecting minimal residual disease, tiny amounts of leftover cancer cells using PCR technology, in this case in a leukemia model.
So like checking if any embers are still glowing after putting out the main fire.
Precisely. It suggests we could move towards personalized monitoring after treatment to catch any potential recurrence really early and step in again if needed.
Wow. So it's initial tailoring plus ongoing personalized surveillance. That feels like a real paradigm shift.
It really is. And these ideas extend beyond cancer, too. Think about drug metabolism.
How our bodies process medications.
Exactly. Our genes play a huge role here. Some sources highlight how variations in the gene's coding for drug metabolizing enzymes can make a big difference. Someone might break down a drug really quickly, needing a higher dose, while someone else processes it slowly and might get side effects at a standard dose.
Right, so knowing that genetic difference could directly influence the prescribed dosage.
It could, yeah.
Hmm.
And another angle is designing drugs, prodrugs actually, that only become active when they encounter specific transporters on cell membranes. The preclinical handbooks mention this. So you could potentially tailor a drug based on someone's unique transporter profile.
Making sure the drug gets activated in the right place for the right person. It feels like everything is becoming fine-tuned. What's driving all this progress? Technology must play a huge part.
Oh, definitely. Artificial intelligence, AI, is a major driver.
How is AI helping here?
Well, AI algorithms are incredibly good at analyzing massive data sets. We're talking huge amounts of genomic data, clinical trial data, patient records. AI can sift through all that complexity, identify potential new biomarkers, and even predict how different patients might respond to treatments. It's connecting dots in ways humans just can't, you know, manage on that scale.
So AI is like the ultimate pattern finder in all this biological data.
In a way, yes. It's accelerating the discovery process for personalized approaches. And things like the FDA's Breakthrough Devices Program, which is mentioned in the AI Source 2, help speed up the review and approval of these innovative tools, getting them to patients faster.
So that's good to hear. The regulatory side is also trying to keep pace, but it sounds complex. What are the challenges in actually getting these therapies from the lab to the clinic?
It's definitely complex. Translating all this genomic knowledge into reliable clinical practice takes a lot of work. Rigorous validation is key, and the fundamentals of drug development still absolutely apply.
Like basic safety testing.
Exactly. Preclinical studies, including things like toxicokinetics, basically understanding how the drug moves through and affects the body, are still crucial. We need to understand exposure and safety, especially when individual genetic differences might play a role.
Makes sense. Even a targeted drug needs thorough safety checks.
Absolutely. And then you have clinical trials. The different phases are essential for evaluating safety and, importantly, efficacy in actual patients. And with precision medicine, these patient groups in trials might be stratified, grouped based on their genetic profiles or specific biomarkers.
So the trials themselves become more targeted.
Potentially, yes. And as we mentioned, those biomarkers can often be used as efficacy endpoints in the trials themselves, direct measures of whether the treatment is working in that specific group.
Using the biomarkers to see the effect directly in the trial.
Okay.
And the overall regulatory framework has to adapt too, I guess.
It does. Regulatory paradigms are evolving to handle the specifics of precision medicines, which often don't fit the traditional mold perfectly.
Now, you mentioned we have some sources from OPR&D, Organic Process Research and Development. How do they fit into this precision medicine picture? They sound more like manufacturing details.
They are, but that's actually really relevant. While specific papers, like the ones from 2020 or 2021, might detail the synthesis of a particular molecule that isn't explicitly a personalized therapy itself, they show the incredible level of detail and control needed in pharmaceutical development and manufacturing.
The nitty-gritty chemistry and engineering.
Exactly. And that extreme rigor, that meticulousness, is absolutely paramount for precision medicines. If you're creating a therapy tailored to an individual's genetic makeup or a very specific biomarker profile, the manufacturing has to be incredibly precise and consistent. There's often less room for error.
So the foundations of good pharmaceutical development become even more critical.
I'd say so. Even something that might seem routine, like using activated charcoal to remove impurities during synthesis, mentioned in a 2023 OPRD source, highlights that commitment to purity and precision. That level of quality control is fundamental, perhaps even more so when you're dealing with highly specific, potentially individualized therapies.
Right, you need absolute confidence in the product when it's that targeted. Okay, so let's try and wrap this up. It feels like the core message is that precision medicine, powered by genomics and biomarkers, is shifting us away from that old one-size-fits-all model.
Definitely. It's about leveraging our deeper understanding of individual biology to create treatments that are hopefully more effective and maybe safer because they're tailored.
And this deep dive hopefully has given you, our listener, a clearer view of the concepts and some of the real-world science making it happen.
That was the aim, to cut through some complexity and show the direction things are moving.
So for a final thought, given how fast genomics and AI are moving, what does the future hold? Where could truly personalized healthcare go?
Mhm.
Are we heading towards treatments that aren't just tailored initially, but maybe dynamically adjusted, almost in real time, based on your changing biology?
It's a fascinating prospect, isn't it? Healthcare that continuously adapts to you as an individual.
Definitely something to think about.